a transition in this course to the applications component of the information that hopefully has provided you with some of the fundamentals with respect to biostatistics and research design. And we're going to start essentially pre-marketing, although the topic itself that you're going to be hearing about from Katrina Rogers has implications beyond the pre-marketing period, and that is the topic of CMC. And it's really my distinct pleasure to introduce Katrina. I'll say a few things. I know I encouraged her to talk with us at the beginning about her journey and her work, but I will start by saying she has over 25 years experience in discovering, developing, manufacturing medicines and related technologies. She has expertise and qualifications in the area of quality, good manufacturing practices. She's an adjunct faculty member at the WSU College of Pharmacy and Pharmaceutical Sciences. And for those who don't know me, that's my alma mater. She is active in the local bioscience community there in Eastern Washington. And most recently, she became the Chief Executive Officer of Evergreen Bioscience Innovation. That hopefully you'll tell us a little bit about that, because we have always a keen interest in understanding this expanding life science ecosystem. So she has her own consulting practice. And I don't know how to better describe you. I think you're a change agent too, but I'm going to walk us through that. So please take it away, Katrina. Thank you so much.
Great. Well, thank you, Andy. It's my pleasure to be here to speak with your class. I'm always very excited to have a chance to talk to people who are in the process of increasing and expanding their knowledge and expanding their capabilities within our ecosystem. So we'll love to share as much as you're interested in about my background too. So let me go ahead and get started on the lecture here. Everybody hopefully can see my slide deck now. From my end, it looks like, okay, good, we got a thumbs up, excellent. So I want to focus today on what regulatory affairs professionals need to know about chemistry, manufacturing, and controls. And you'll see as we get in here that I'm stretching this metaphor a little bit here to cover kind of the three different spaces that we typically look at. But I can do that because there's a lot of related information. And I think you've seen that over the past year and a half of your training. And hopefully the metaphor won't be stretched too far. But Andy wanted me to talk a little bit about myself. So I have industry experience. I started my career at Pfizer after getting a degree in chemistry at the University of Washington. And I moved on from Pfizer. I was in drug metabolism at Pfizer. I moved on from Pfizer to work in drug metabolism at a contract research organization known as MDS Pharma Services. Advanced through there to become a study director and technical director for pharmacology and metabolism studies at MDS. And then moved back to Spokane, which is my hometown, to take up a role at sterile medicines manufacturer Jubilant Hollister Steer in the quality unit. So that's kind of the space that I've been operating in. A couple of years ago, I had an opportunity to join Washington State University as adjunct faculty. And as a faculty member, I have been the PI on a couple of talent development grants, specifically focused on helping introduce young people who are in high school and just post-high school to life science careers. And then I'm also an entrepreneur, as Andy mentioned. I have my own professional services firm. And really, in that role, I help people who are innovators, who are investors, and who are leaders get their medical products from bench to bedside. And that can involve a wide variety of roles, wide variety of work, consulting on regulatory pathways, getting people connected to sales experts or other folks that they need to, as well as contract service organizations that they will need to do their work. And I'm also the CEO of Evergreen Bioscience, and that is a whole presentation in and of itself. We're a modern innovation cluster forming here. We're based here in Eastern Washington, but we aim to make the Pacific and Mountain Northwest a magnet for companies that consume and provide contract services for the pharmaceutical and medical device sector. So we feel that because contract services are so fundamental to the work of what we're doing, that by elevating the companies that we already have in our region and attracting and growing new companies that fill the different gaps, we can make our overall ecosystem much more robust. So excellent. So now that's enough, probably enough about me for now, but I'll be happy to take more questions later. Let's talk about you guys instead. Here we go. So as I understand it, you are almost to the end of your master's degrees in regulatory affairs, and I want to congratulate you. It's an undertaking that is, I know it was time consuming. You've learned a lot. There's quite, I think probably what you've learned as well is that there's quite a bit more to know than you had a chance to be exposed to, but it's an achievement and you should be very proud of it, and I congratulate you. However, one of the things that you've been exposed to in this course are some of the roles that you might be expected to play. Things like logistics, regulatory affairs, but also you may work with the quality unit, you may work with the quality control unit. You're likely to work with senior leadership and finance at some point. And all of those roles are really focused on the discovery, development, and commercial manufacture of medicines and medical devices. So what you don't know about, or you know less about at this point, is how these materials, right, the medicines and medical products that have been discovered, how they get through development into a commercial space. And that's the chemistry, manufacturing, and controls piece of it. So everything from those early stages of manufacturing, right, the drug product or the drug substance and the drug product, as well as all of the related functions, the distribution, the QC lab, the quality assurance group, as well as the labeling and packaging group. These are all topics about which you have had very little exposure up to this point. You may have had some exposure if you came to this work or to this degree program from a role already in the industry, but not everyone has a chance to touch all of these different points. And more importantly, you want to have a sense of the bigger picture, because as a regulatory affairs professional, you are going to need to and want to communicate to your supervisors, your employers, as well as to the inspectors that might come to visit your organizations, information about how your processes manage the safety, the efficacy, and the quality of the products that you're producing. So in this presentation, what I hope to do is show you a little bit about the chemistry, manufacturing, and controls for drugs, biologics, and medical devices. And I've kind of changed this up a little bit by saying the chemistry piece is providing a quantity, making the product is the manufacturing piece, and the controls piece is really about ensuring the consistency as you make those products over and over and over again. All right, so I do want to mention that if you have any questions, Nikita's paying attention. She will, you can either drop them in the chat or raise your hand, and it can often be a good idea to ask questions as we go. So don't hesitate to step forward. All right, well, let's go ahead and get started talking about providing that quantity, the chemistry piece. And I've broken this one out into the 3 different sections, and you'll see that I've got it, I have little codes for the different sections. As we go forward, the sections will get a little bit shorter because there'll be a little bit more overlap. So when we talk about drugs, and we're talking about small molecules here, we think about the drug substance. What is that upfront piece that is the active material? You probably have seen a graphic much like this in other courses as well. I've simplified this quite a bit so that we can focus on where the chemistry, manufacturing, and controls come in. These are the stages of chemistry piece that occur for the small molecule. So we'll see selection and characterization in that discovery to toxicology phase. We'll see formulation and analytical work being done in the toxicology to clinical phase. And production really starts in toxicology because you have to have that robust drug substance supply in order to supply the large studies for safety for toxicology, as well as you get into the clinic, you need those full-scale supplies that are going to be relevant to what you eventually will commercially produce. So we'll talk about each one of these by themselves, right? So selection and characterization is really about scaling up the drug substance. So what happens is we start out with a molecule that's up there on the left-hand corner, and it's a complex molecule. And quite often the medicinal chemists will have worked very hard to find this active material, but they only have one pathway to get to it. And that pathway may have multiple steps, and it may use very expensive materials, and it may be very low yield. So there are special medicinal chemists who work just on making sure that they can reduce the synthesis complexity, reduce the number of steps, reduce the cost of the steps, and increase the yield. Because that ends up being a business decision. If you can't get it to scale it up, you might have to kill the program if you can't find other active material. Those chemists will also work with other scientists to improve the solubility, the pourability, and other physical properties by choosing different salt forms or processing the drug substance in a slightly different way. Next, the next step in that chemistry piece for small molecules is thinking about the formulation and the analytical piece. So for formulations with small molecules, you may be in a capsule or a tablet. The ideal, of course, is an oral dose form. But some molecules are just not amenable to those kinds of modes, and they need to be made as sterile injectables. So those are two types of formulations that would be typical for small molecules. At this point, scientists in the companies are also thinking about how to form the active material as well as the placebo in a formulation, because long-term in clinical, you need to have the placebo be as identical as possible to the active so that the blinded trials can go forward confidently. And again, this occurs in the toxicology and clinical stage. You may not need an oral formulation, for example, for your tox studies. The dosing may be quite simple there, but you will ultimately want that final formulation as close as possible when you start to move into the clinic. You'll also need to worry about making measurements. And for the drug substance, we need to measure its potency, we need to measure its impurities, and we need to measure any other parameter or physical chemical parameter that is required to maintain its quality. So that might mean moisture level, and it might mean overall, it might mean sterility or a certain amount of low microbial, what the FDA likes to call no objectionable organisms. And this is an example, there's an example of some of the testing that might happen here where we have a chromatograph up above, and then a chromatogram shown in the picture below. So there's a whole set of scientists just devoted to working on the analytical piece for these small molecules. And for production in drug substance, the context here is synthesizing phase appropriate material. Just as we had a team working on taking the molecule synthesis from many steps to fewer, we also have a team, and it may be the same team, that is working on scaling that synthesis up to a multi-kilogram scale. So early discovery, the syntheses will typically, if you have 100 milligrams, you're probably thinking you're doing really well. To get into the larger tox studies, and if you're going for non-human primates especially, you will definitely need multi-kilogram. Excuse me. So by doing this, we go from on the left-hand side, that hand synthesis, into the large production facilities where they're using 300 to 500 liter tanks and making very large scale syntheses. So in the chemistry part for small molecules, we have the selection and characterization phase, we have the formulation and analytical phase, and then we have the production to make the active chemical material that's going to go into the dose form. So what does this look like for biologics then? And for biologics, I like to call it, it's still drug substance, but I like to call it the active ingredient separately because that's a little bit different. So we see the same stages for biologics. They're just, again, a little bit different because of the source of the material. So in early discovery, typically the biologic is produced by cells, and in almost all cases, it's produced by some sort of microbial cell. And it will be produced at a laboratory scale in a flask culture, and often a very small scale at that. So in order to get, to targeting the active ingredient scale, scientists have to think about what kind of cells will be used. Do we use a bacterial cell, which is very good at growing and very good at not contaminating the final protein product with adventitious viruses and endotoxins? But it also, bacterial cells have trouble making some kinds of proteins. So people will choose mammalian cells like CHO cells or other types of cells that can be grown to a larger scale. But you often need to figure out which cell is proper, how to get it transfected to make the proper protein, and then ultimately, what scale do you really need? If you're having to make, again, multi-kilogram doses, you're going to need to move from those flask culture devices into potentially a 15 to 25,000 liter size in order to of bioreactor in order to get the active ingredient at scale. Once again, suitable dose forms are very important. For biologics, there are very, very few of them that are oral, though there are a few. Most will be administered as a sterile injectable, either formulated in a small vial or formulated in kind of a pre-formulated into an IV bag type of situation, a liquid. So they can be either liquid or lyophilized depending on the parameters of the biological. Again, analytical is very important here, but the kind of analytical that is done is usually different. We see less chromatography in these cases and more electrophoresis and gel blots, like the Western blot shown here. And we also see ELISA tests, which are another kind of immuno test. And those are commonly done on automated systems like the one shown on the slide. Lastly, for production, we are focused on producing that bulk phase-appropriate material. And it's true for both drugs and biologics that you're looking for phase-appropriate material. So you may start with, this process of scaling up the production will start with things like an engineering batch, which is a really early stage. And it's not done by GMP, it's done with an SOP, but the SOP is often marked up. Then you move on to a demonstration batch. And then from the demonstration batch, you're looking at the process qualification batches, and then moving towards the process validation batches. So there's a set of steps that you go through. If you can use your engineering or demo batch material for your toxicology studies, if it is of good enough quality that the demo, the late demo or the early PQ can be used for the clinical stages, you're saving the company a ton of money as well as a lot of time in terms of the process to get the therapy to market. Multi-kilogram scale are here on the left three images show a multi-kilogram scale. On the absolute left, we see a tank of the sort that you might see in a larger production facility. We see a person in a bunny suit operating the automation for the bioproduction, as well as we see some isolators in here too. It's an isolator line with where the line is all separated from the operators using these glove boxes. You may also be engaged in working in personalized medicine, and personalized medicine means things like CAR-T or cellular therapies. Seattle is the home of really the founding home of a lot of that work. And so you may end up working with a company that's doing that. That's the picture on the right, where one or two people are working with a very small set of equipment to make a personalized medicine out of plasma that has been withdrawn from a single patient. So the scales can be really large or they can be kind of one person. So again, in the biologic space, just like as in the small molecule space, we have a selection and characterization. We have a formulation and analytical, and we have a production phase. It's just the activities that are different that happen in those spaces. Excellent. How are we doing on time? Not too bad. Okay, so let's go on to the devices. And for devices, we're going to take a kind of a right-hand turn here, because devices, your early stage is really about the viable design. So in the device stages, we think about the requirements for the design, which drive the design. We think about the components and assembly, which then drive more of the parts of the design. And then we think about, obviously, production, which is really about producing the device. So the requirements phase is really critical. You have to understand what must the device do and who is using it, because it ultimately leads to the questions around how easy should it be to use, how cleanable does it need to be, and what's the risk of injury if, for example, an inexperienced user is using it. So you could see by the pictures here, we might have a device that would be used in an operating suite, so where cleanability would be very critical, being able to sanitize and sterilize it. The workers there are a lot more skilled. They're going to be trained, so maybe ease of use you can back off on. There could be high risk of injury if it fails, so that you have to think about what are the failure modes for a device. For a home health monitor, a blood pressure monitor, that's a home monitor. It needs to be really easy to use because people are going to mess up with it. Probably should have some cleanability, but again, it's at home. It's a single or two-person use, so it's good to have it be able to be wiped down, but it doesn't need to be sterile. And the risk of injury does exist. So again, the failure modes that you have to think about is, what if an inexperienced user is using it, or someone like a child gets a hold of it and starts to use it. As we move on to components assembly, it's important to think about what the manufacturing techniques are for the device, as well as the materials. If you're making it from metal, you're going to use a different technique than you would from polymer. You have to think about how many parts are involved. The more parts there are involved and the more fiddly things that needs to be assembled, the more expensive it would be. So it's important that the design tries to move in a direction that will use fewer parts, easier materials, the cleanable materials or the sterilizable materials if you need them. And then thinking as well about the protection that the users will need in terms of, and the assemblers will need in terms of making the device. Production is focused on how many are needed at each step. When you go into bench testing, maybe you need one for bench testing, or two if you have two contract providers and you send them each a device. When you go to clinical testing, you're going to want enough to send out to your key opinion leaders and the folks you have recruited for your clinical testing. So maybe that's 10 to 100. And when you get to commercial scale, it really depends on the device. If it is a small diagnostic, like the COVID devices shown here, you're going to need thousands, if not millions of them. If you are working with a device that is complex and is used only in certain operating suites, maybe still you might only need 100 to 1,000 of them. So again, for devices, just a little bit different here. The design is really important. So requirement setting to begin with, understanding the components and assembly that you're going to need to do to make that design effective, and then working through the production issues so that you get your design right before you need multiple of the devices.
Katrina, what is that picture at the bottom?
The picture at the bottom is a orthopedic device being 3D printed. So if we go back up here on the left-hand side, we see that's a typical, like a titanium machining unit. And then there's that picture of the orthopedic device at a little bit larger scale being 3D printed.
Great, thank you so much.
You're more than welcome. Okay, so let's move on from the chemistry piece, that early stage, right? Or the, I guess, the front end of the chemistry, manufacturing, and controls, and talk about manufacturing, which I like to think of as just making the product, right? So this is what will actually come out and be used by the healthcare provider or the patient. So for the drug product, so we had drug substance to begin with, we have the drug product now. Because drugs and biologics start to get very similar when you get into the drug product stage, I've combined those two. So this should go just a little bit faster than some of the previous sections. So here we have our timeline again, and our drug product stages start a little bit later in the timeline and then go. I think one of the things I want to make sure that you understand is that the production pieces will go throughout the lifetime of the product. So that all of these things will end up going into the long-term file and maintenance of the product. So for a drug product, we have a facilities phase, we have packaging and labeling, and we have the process piece. So the facilities is really about where you're going to manufacture it. What are the requirements from the perspective of, what kind of quality of water you need? How clean does the facility have to be? What kind of warehouse storage capacity do they need? What is their inspection parameters, and what are their inspection equipment that they can use, as well as laboratory equipment and staff? And then the production equipment itself, that's shown in the upper right-hand corner. So it's kind of left to right: water, cleaning, warehouse and distribution facilities, the inspectional equipment, the laboratory, as well as the actual manufacturing equipment itself. Quite a bit, as I mentioned earlier, quite a bit of this work is done by contract manufacturing organizations. And over 60% of new therapeutics are formed in small or virtual companies. And so if you're working with a smaller virtual company, if you are an employee of a smaller virtual company, you're going to be working with commercial contract manufacturing organizations. So it's kind of important to understand that they each have their own specialties, and they will each be suited for different types of products. So as you can see, the selection of the facilities is very important in that toxicology to NDA stage. You will file with the specific facilities that you have picked out. You can add facilities or change facilities in the future, but it's going to take a filing change. So it's always good to be thinking about this up front. You also will want to be thinking about where the distribution is, because if you're just in the United States, maybe you only need domestic facilities, but you might need facilities outside of the United States if you want to distribute the product to other countries. The next part of the process, the next part that's very important is understanding whether or not the process can actually be validated. So by validation, what I really mean here is, can we take a set of incoming ingredients that have already been certified as quality materials? Can we formulate them into a drug product? Can we then fill that drug product and then to create the final dose form? And then that dose form needs to meet its specifications, the quality parameters that were set for its final form. And that's potency, purity, could be sterility, almost certainly has some kind of microbial measurement to it, as well as all of the other parameters that have been set to show that it is a quality product. And the validation piece occurs over multiple steps with, again, the demo batches, the engineering batches, the process qualification, and the process validation batches. These typically occur as you're going forward. PVs will not typically occur until the product is about to be approved or has just been approved. So the very first commercial batches will often be PV batches. For packaging and labeling.
What was that? I'm
Andy,
sorry. What was that acronym you just used? PV or?
Process validation batches. PVs are process validation batches.
Thank you. Thank you.
Great question. Because process qualification batches are PQ batches. For packaging and labeling, we need to think about what the final product presentation is. And this is where I know that packaging and labeling is a place where a lot of companies, there's a lot of regulatory work involved in changing packaging and labeling and updating packaging and labeling based on new information that might come from the post-marketing studies. It's the information that's on the label is important for the end user, and the end user determines what kinds of information will be on that packaging and labeling. So if you have a bottle of, as on the left, packages have to include the dose form, they have to include the name, they have to include the both the trade name and the generic name of the material. They have to have instructions for dosing and use, and that typically comes as part of the leaflet. And I've shown you an example of the leaflet on the right-hand side that comes in that instance, that is a birth control medication. If it's over the counter, there's a whole separate set of information that kind of has to go through. And you, again, it's very fit for purpose, you know, depending on who is the end user. So for drug product, and this is that manufacturing phase, we are looking at assessing the facilities and making sure we have the correct facilities to make what we're trying to make. We have a process that can be validated to produce the product that is the desired target product. And we have packaging and labeling that assures us that the product inside is what it purports to be. So again, devices a little bit different here. We are looking at, in this stage, at what it will be the commercial product. But again, the stages themselves are pretty similar. You have to fit the facilities for purpose. You're looking at the process to manufacture it and making sure it's reproducible, and then making sure that the instructions and packaging don't get your end users into trouble. Device facilities are very dependent on the output. This is a citation from 21 CFR Part 820, which is this is the quality system for medical devices. And it points out that manufacturers will only need to comply with those requirements that are applicable to the device that they're trying to make. So on the left-hand side, here's a device. This person is inspecting a device that has to be maintained in very clean conditions. So they are in a clean room environment. On the right-hand side, the technician is working or assembling a device that does not require that kind of cleanliness. And so they can work without gloves, but they do have correct eye protection as well as correct clothing protection, which protects them, but also the device. That process is still going to need proof. You're still going to have to be able to demonstrate that your manufacturing process, you can take those parts that you've determined to meet the quality parameters that you want. You can put them together in a consistent way to make a final product that meets the original design that you have submitted. So maintaining the design in a medical device is very similar to maintaining the file or the submission file for a drug product, maintaining the quality and the consistency of that drug product. And the final presentation is ultimately also part of that design file. And I've showed you a couple of presentations here. In the lower right, we've got a package for a particular screening test, which has its instructions. It has all the materials together. Each material is separately labeled and carefully, the colors and the combinations are carefully put together so that it's not confusing for the end user. On the left-hand side, we see a label, and what's interesting about this label is it shows an example of a unique device identifier. This is a requirement that the FDA has implemented and regulatory agencies around the world have implemented for devices, because quite often the devices come in a range of types, but most can be identified uniquely in this way, and then that allows traceability back to the original batch that they were manufactured in as part of, as well as it provides good traceability for manufacturers if they have devices that they can bring back and recondition, such as an instrument of a diagnostic instrument or some sort of instrumentation for surgical use. So again, the unique device identifier, that's a quite a unique thing for medical devices. And then the instructions and packaging are all put together to help the end user not become confused and to be able to use the device properly. So for the manufacturing piece, again, with the devices, the focus is on facilities, a consistent process, and the instructions and packaging that all work together to make sure that the devices that come out meet their design specification. Great. All right, we're doing pretty good here on time. We're on our last section. Let me take a quick drink of water. Thank you. So controls is our last part, and this is all about ensuring consistency. Whether you're working on a medical device or a therapeutic, a drug or a biologic, it's the controls piece that you're going to be following and that the agency is most concerned about, because that's where you demonstrate that you can consistently manufacture over time. Because this is so similar, this is going to be broken up into different chunks, and we'll talk about the drugs, biologics, and devices kind of all together in this section. Part of the controls is your supply chain. I know we hear a lot about that word these days, but for the CGMP manufacturing, those requirements apply to all parts of the supply chain. And so that's everything from the very early stages of the materials that are coming in, all the way through the different parts and pieces that you're assembling, and as well as your ultimate distribution channel. You will be engaged quite heavily in the supply chain piece for your employers. It's an important part of the quality assurance unit, but the regulatory affairs folks get involved quite a bit as well. How do you ensure that the CGMP requirements are being applied correctly by your partners, by your suppliers? You'll be conducting supplier audits. There'll be an incoming inspection process that's typically managed by the quality control unit and then also overseen by the quality assurance unit. And there'll be change control. And when these suppliers will need to inform you of any changes that they're making, and those changes might affect the filing, and you'll have to make changes and submit reports. You'll want to also make sure that the suppliers are doing proper deviation investigations and implementing the immediate corrective and preventive actions that are a correct outcome of those investigations. I will tell you that the deviation investigations and the CAPAs are one of the places that regulatory inspectors will look very closely at, because that's one of the #1 things that they find when they're inspecting is people aren't doing the proper investigations or following through with their CAPAs. And here, as in almost everything, but certainly with CGMP supply chain, relationships are vital. Your finance folks will want to make changes because things are less expensive in some parts of the world where they are in other parts of the world. But those changes could have a significant impact on your product, and they have to be managed very carefully. So it's much better to have long-term relationships with solid suppliers who can help you achieve your goals than it is to hop for cost purposes from supplier to supplier. The quality systems of your facility will be a critical part of maintaining control. And I'm sure you've already talked about the 6 quality systems that are shown in this little diagram here. We'll take a look at some a little bit more here. The quality system is a fundamental requirement for drug products. And the guidance, one of the things that I've shared with you for your reading material would be this US FDA guidance, which is their quality systems approach. It's a 2006 document, but it's still very relevant. The key elements of a good quality system, management oversight is the first thing they're looking for, right? There needs to be leadership, and the leadership has to be maintained. The quality unit needs to be separate from the manufacturing unit. This is to prevent a process from taking on a life of its own and the business risk outweighing the quality risk. Your investigations and CAPAs system will be very important. Reporting to the agency is also critically important. that you have good documentation, procedures, and records, and change control. All of these things work together to make your production system, your facilities equipment systems, your laboratory controls, materials system, and your packaging and labeling systems working together. This is the quality system gives you the evidence that everything is under control. Similar in device quality systems are similar, but they're not identical. And the differences tend to be right around the kappa space. So when a deviation occurs, rather than opening an investigation in a device quality system, you open a kappa, and the kappa holds both the investigation as well as the ultimate corrective and preventive actions. And those deviations might come from reports of corrections and removals, or from field reports, or from your tracking of your medical devices through the UDIs. Similarly, in device quality system, one of the things the agency will be investigating is your design controls. Are you maintaining the products that you're producing according to their filed design? We have the material records documents and production and process controls as are typical for a pharmaceutical, as well as facility and equipment controls. And I changed up the reference a little bit this time. What I'm giving you for your reading assignment is the inspectional reference, Guide to Inspections of Quality Systems for Medical Devices. So the FDA publishes procedure manuals for their teams, their inspection teams, and these are full of really valuable information about what they're going to look at, why, and what their process will be. So I wanted you to get a chance to take a look at that because if you're working for a device company and you're going to be supporting an inspection, you'll want to know what they're looking for so that you can give them the right information. Lastly, we've got process controls, and Dr. Strigachis wanted me to talk about statistics, and this is where you'll see it quite a lot. But basically, your process controls are really focused on the recording of data, and good documentation practice is very vital for both medicines and medical devices. You'll need to keep training records. You've got to have batch records. You have to have a master batch record, and you have to have individual records for each batch that's produced. You need records of the testing that match up with the batch records. You need to investigate any deviations, and those have to have records. And then you have to have overall the quality records that demonstrate that you're following all your processes correctly. The data from those records will inform your investigations. It will also inform the annual reports that you are required to send the agencies to demonstrate that you are operating appropriately. Similarly, that data can be very valuable if for some reason a deviation shows that a process change may need to be made. And you'll see lots of control charts. These are super important. You'll see them for incoming materials. You will see them for stability. You will see them also for the production over time and in annual reports where you're reporting on the batches that have been produced over that annual period. So what I hope that I have given you a flavor of by stretching the metaphor a little bit, how the chemistry, the manufacturing processes work with all the allied processes, such as quality control, quality assurance, and distribution, as well as packaging, so that you can have an overview, a sense of what you're doing, and how your company is using the systems that it has to ensure the efficacy, the safety, and the quality of the products that you're producing through chemistry, by providing a quantity, manufacturing, making the product, and controls to ensure consistency. And that's the end of my slides, and I'd love to talk with you now.
I encourage you all to unmute and ask questions. I think what we'll do is after a period of Q&A, maybe take a break, and Katrina and Nikita will go ahead and publish the homework assignment for this session. and maybe ask you, Katrina, to sort of walk us through that in terms of, you made reference to the readings. They were actually embedded in the document with the homework assignment. So I, for one, think it would be well worth spending a few minutes after the break, having you walk us through the homework assignment and the expectations there. So let's go ahead with Q&A now, and then take maybe a 10-minute break or 15-minute and come back at unless we go longer with questions and answers.
Great.
I can call on people too. I know it's late in the day.
I guess I just had a question about how you were discussing the statistical point of view for process control. So I guess my question is if you could give a little bit more insight and background into how statistics plays into that piece.
Okay, this is an excellent question. Let me pop back here. So what we see with let me get to that slide too, because I think that will help a little bit. Okay. So one of the things that we quite frequently see would be control charts. And there's an example shown here where we've got percent recovery and the samples, and you can see that there's an upper control limit and the target, the control line, which is the target concentration for recovery. And rather, the target is typically 100%, but the control line is where the average turns out to be. And then you have a lower control limit. And then if you see the yellow lines, these are the statistically calculated actual limits of the data that is shown. So what happens when you're making batches, and so typically you'll make, as I mentioned, right, there'll be 3 process qualification batches, and those will go on stability. And then you'll have 3 process validation batches. And so each of these batches will be tested they'll be tested for release, and there'll be data for that. And then they will all 6 batches will go on stability as well, because you need the original stability data from the PQ batches to demonstrate that to provide yourself a shelf life. And then you use the PV batches to validate that shelf life. And then over time, after the product has been approved and you've been manufacturing it for 2 to 3 years, you're going to end up with, depending on how often you manufacture it every year, you might end up with 4 or 5 batches, or you might end up with 20 to 50 batches. Just really depends on the demand for your product. So you might have many more batches of things like a vaccine, where a product that was only going to be used in maybe 100,000 patients every couple of years and might have a long shelf life. Maybe you only need to manufacture a batch or 2 a year. And so the control charts, you use the statistics. There are well-known techniques to calculate both the control, how it's actually behaving versus the specifications, which are what is typically monitored as the upper control limit and the lower control limit. Your specifications are often set by the data from the PV batches, though it's usually wise to kind of be a little bit more flexible about what your specifications are to start with, because you may be able to meet a very tight specification with a few batches, but it gets a lot more tricky when you get many batches, as are shown here, where you get a lot more variability in the particular data that is that data piece that you're looking at. It's possible to set too tight a specification, let's put it that way, and then end up with a filing that really causes you a lot of trouble. Melissa, was that sufficient an answer?
No, that was great. Thank you.
You're welcome. They're all stunned, Andy.
Yeah, I'm assuming that statisticians are brought in to do this sort of thing, or is that part of, let's say, a regulatory affairs professional's duties under other duties as assigned?
Well, that's one of the things that a regulatory affairs well, I think the regulatory affairs professionals seldom make the charts. But I would say that the people who are making the charts are rarely statisticians. Will use because these the calculations are fairly well established. An ordinary practitioner can learn to use them appropriately, and in many instances there'll be an SOP. This is how we want you to chart this is how we want you to do the work. I think it's rare for an innovative company might have some statisticians on staff to be able to work on that, but I think it's really rare for most companies to have a statistics professional doing this work. It's typically the quality professionals that will do it.
Okay. Very helpful.
Katrina, we have a question from one of the students in the chat, and they want to know what is the most challenging aspect for the regulatory team with respect to CMC?
Some people will say that it's actually it's the filing, but I think that is not. I think the initial filing is fairly straightforward. I think what is the most challenging is maintaining the file over time, particularly if there are a lot of if there's a lot of manufacturing and there are a lot of minor variables, right? If it's not well controlled. So when the agency always encourages us, similar to medical devices, but also for therapeutics, they encourage us to investigate the design space around the actual manufacturing part, the chemistry and the manufacturing part, so that we understand what minor changes to the process, what impact those might have on the product. If the development team has not done a good job of investigating that design space, when you get to the commercial phase, any change that occurs, you may have a lot of trouble justifying release of the batch because you don't have the design space information to cover it. So in other words, maybe you don't understand what will happen if, for example, the storage temperature deviates by 3 or 4 high degrees too high during one part of the manufacturing process. If you have a change in a starting material, you don't understand what might happen. If you put in too much or too little of a starting material, maybe you don't know what might happen out of that. So those are cases where you get a surprise deviation because it almost always comes out in the quality testing. Why is this different, right? We've got a result that doesn't match the specifications. Then you have to go back through the investigation to find out what happens. Similarly, if I think if you justify a lot of, if you have some design space and it's already built into your filing, then there's a lot less changing that has to happen going forward. That was a great question.
Other questions for our expert?
Hi, Katrina. I have a question regarding about kind of like scaling up. So I work in a personalized medicine of a manufacturing facility, and scaling up is always kind of like the topic on the table, right? How we're gonna make more money. So can you share some experience when you're like going through scaling? What kind of like challenges you guys will run into?
That's a great point. And I think cellular therapies it's going to be interesting to watch that field as it goes forward, because right now they're being manufactured one patient at a time. There are some startups working on more genericized therapies, making it more accessible using a more generic technique, but they have not yet gotten into any kind of proof of concept studies. I think ultimately for cellular therapies, the challenge there is really reproducibility. So the same process that you use, because the starting material is different And because some of the pieces of the process are very specific to the operator or very specific to, it's very hard to standardize, and the quality parameters and the reproducibility is not quite there yet. So it's very unfortunate that what we see is those therapies are quite expensive, partly because it takes a lot of very skilled operators doing very careful things over time, but it's also hard to reproduce even when you have those situations in process. For a biologic or a small molecule that's, say, manufactured in a tank or using a synthetic method, the scale up tends to be more straightforward. However, you always run into problems. So and some of these problems can really actually kill a program. If you don't have the right active chemical material, you can't get it to scale up. The challenge there is to really make those processes robust enough so that you don't lose a batch, because losing a batch often incurs a huge cost in opportunity time, but it also incurs a tremendous cost in terms of early materials, the materials that are used. Ben, did that help answer your question, or do I need to clarify a little bit more?
No, that's perfect. Thank you very much.
You're welcome.
And kind of like adding on to it, sorry, keep asking questions. So when you're talking about like a little bit more larger scale, right? Like I'm kind of thinking about like, what is, if I were to like, what's the transferability in terms of skill? Like, do they have relatively similar requirement for skill? So for example, if I work in a small biologic manufacturing site, and then if I move on to a large scale, what kind of skill that's a little bit more transferable?
Right. Well, the basic skills that are transferable is really understanding that these are processes that have to occur by the CGMP. So the documentation, the records, the requirements for training, understanding that there are requirements out there, having that expectation. All therapeutics have to go through a stability program. That's what determines their shelf life. So you know that there has to be quality control testing that starts at the beginning of the production, that starts at release of the batch and then goes over time to show that the product can continue to meet its requirements over a certain period of time. You always know that that both biologics and small molecules, they go through an NDA type process, something at that, it's either a biological license application or an NDA for small molecules. Those are similar in the context. They may not be exactly similar in the content, but they're going to be close enough that you can make a good mental map between those two. And similarly, a lot of the incoming processes are very much the same. So the production may be slightly different. And again, when you get into drug product, the production pieces are so similar that it's quite easy to transfer back and forth between those two regimes.
Gotcha. Thank you very much.
You're welcome. So we've got something else in the chat here.
We have a question about QC tests and a sample from the batch.
Okay. Larissa, I thought about putting a question about sampling. Sampling is a thing that blows everybody's mind. There are, the industry uses ANSI sampling procedures. The sampling procedures are different, whether you're looking at attribute sampling, which is the attributes are things like its appearance or its color, or whether you're looking at more of a quantitative type of testing where you're measuring something within the product. It's the number of samples that significantly represent that batch that has an impact on how many samples you take for quality control. It has an impact on how many samples that you take for your inspection process, and how many deviations that you can see, or flaws, in other words, that you see in your inspection. All of these things are set by those ANSI sampling criteria. And I couldn't tell you exactly how they operate because it astounds me every time that anybody can actually figure out those tables, but they're dependent on the number of batches that you have, the size of the batch, you know, how much, you know, what is the actual quality number that you're seeking. It's quite complicated.
So if I understand, these tables are available is it part of the regulations, or is it more along the line of best practices that people can access if in the field?
So quite typically, an organization will have the appropriate sampling tables, sampling plans, and sampling tables available in an SOP. They're not provided by the agencies. They expect you to go and find the relevant ones that you have. They can be really quite interesting. The standards, I'm going to drop a link into the chat from the from the ASQ organization that kind of talks a little bit about these sampling plans. But fundamentally, this is a significant, I would say that if there's anything that you're going to use a statistician for, it's going to be the sampling plans. You're welcome. That's a very challenging question. I thought, oh, could I do something in the homework on sampling plans? And I'm like, no, because I couldn't write the key.
Any other questions or comments? Let me ask the class, would you like Katrina to give you a little orientation to the homework assignment as well as the readings now, or do you want to take a 15-minute break? Anyone?
She can go over now.
Okay, look, do we have a second?
Oh, I see another one.
We can go over it now.
Okay, so it looks like we're going to get done a little early today. So I went ahead and published homework number 3. And Katrina, do you want to bring it up on your computer, or do you want me to bring it up on mine and have you just, you know, walk the students through it in terms of expectations and the like?
I have it open here, so
okay. And I think on that same document are the readings, and maybe you can speak to that as well. Okay. Really appreciate it. And then we'll call it. Yeah, we'll see whether there are questions, and then answer the questions, and then we'll call it an evening. Thank you.
Sounds good. Okay, I'm going to share the screen. I'm going to share first. I'll show you the readings, and then we'll go to the Word document that has the links for these. So the first of the readings that I've assigned you is a guidance. This is a specifically written guidance for industry that's published. It's designed to give industry participants a sense of what the agencies are thinking about quality systems in this instance. It's pharmaceutical CGMP regulations, but if you come down here and you'll see that it covers the work of CBER, as well as the Center for Veterinary Medicine, which is quite similar as well. It's entirely possible you could end up working for a veterinary medicine manufacturer. And then it's the Office of Regulatory Affairs typically oversees a lot of the inspectional activities. So if we think about CDER, they're the folks who do the reviews and the approvals for INDs and NDAs, CBER's for biologics license applications, and the CVM for veterinary medicine applications. Guidance are all very typical. They start out with this cover page, and then they start out with yet another cover page that tells you a little bit more about who published it or where you can get additional copies. And then there's a table of contents that really goes into some good detail. And this is a good way to skim a guidance to understand, does it have the information that you're looking for? There are lots of guidance out there, and some of them, you may need 2 or 3 of them when you're working on something. So as you're skimming through, it's a good way to be able to figure out where you're at. This one talks about modern quality systems for pharmaceutical development. and the quality system model for pharmaceuticals. So this is a good way to kind of think about the higher overarching activities of what you're doing before you get into the weeds. You probably won't need to read through all of this to get a really good sense, but it's very useful in the context of the controls piece of your chemistry, manufacturing, and controls. I've also shared with you a link to the Office of Regulatory Affairs inspectional references for medical device manufacturing. So these are the instructions that the inspectors will use when they are actually out and doing an inspection at a manufacturer's facility for compliance. So it's different than a guidance. It's a living web page. They will update it at times. So you can, at the top of the page, they usually tell you when the content is current up here, up in the upper right-hand corner. So this was last updated in 2014. So these are pretty consistent. There are also technical guides linked to a link on this page as well. And those often will dig into in further detail some of the specifics about the wider quality systems guidance. So this one's very focused on quality systems. There will be typically, to start out with, you might have a pre-announced inspection or an unannounced inspection. There will be instructions for the inspectors on how they're to get started and then what they're supposed to do. Typically, when the inspectors come in, they will have a plan. They will know what they want to inspect based on your prior inspection results, as well as any field alert reports or other information and the annual reports that have been sent in. They will, unless they have serious concerns at the start, they will target 1 or 2 of your 6 quality systems to look at. If in the first day or 2 of the inspection, they find an issue with those quality systems, within the 2 that they're looking at, they're absolutely their right to expand the inspection to other areas of the quality system, wherever it might lead. One of you asked earlier about what can be most challenging. Perhaps managing an inspection and responding to an inspection can be some of the most challenging things you'll have to do as a regulatory affairs professional. So, but as we go in through here, you can take a look at each one of these. The links will show what the objectives are. They will be looking, these are the 10 different things that the inspectors will be looking for. There'll be a chart the flow chart that they'll go through to think about, are the decisions being made correctly for CAPAs, for example? This is just a very standardized process. If you understand what they're looking for, you're gonna be much better prepared to think about, when the inspectors show up, what it is you want to be putting in front of them. What are the policies? What are the procedures? What are the processes? And then what are the ultimate procedures that support all of that, and the records that show that your quality system is working properly. So those are the 2. And let me share, those are the 2 reading assignments in their actual thing, in their actual native environment, I guess, if you want to call it.
I have to say, it's funny to see a graphic of an overhead projector.
It really is, isn't it? You're like, some of the things at the FDA are quite behind the times, let's put it that way. So these are the reading assignments. The links are here at the top of your reading assignment document. And then we have 6 questions. So the first 4 questions are easily answered using the documents, using the reading assignments. The 5th you probably picked up while we were talking during the presentation, but it's also on the slide deck. And then question 6 is a fun one. I've given you a figure and a real life example, or I guess a real life made up example of something you might see as a regulatory affairs professional when you're reviewing the annual reports. So I think this is a pretty nice. Nalivaptan is a made up drug name. There's actually a website that allows you to make up drug names for this very purpose. So I thought that was fun to find out. And it talks about the stability program, and it uses this chart for the stability, which again, this is we use some statistics here. We plot the actual concentrations of the percent label claim for the product that were tested. And then we use statistical techniques to construct a trend line for each of those batches. And then we take a look at what the results tell us. And so the questions are really focused on what does this chart tell you about, what would you be concerned about with this chart? So hopefully it will be quite straightforward to answer. And then hopefully that you will have a sense from both the reading and from my discussion what the quality unit should do when in response to these results.
You know what my next question is going to be? Are there any questions?
Now's your chance to quiz me about the quiz.
Well, if you all feel comfortable doing so, start your video and let's say thank you to Katrina Rogers. Again, feel free to ask questions. We still have time. Questions about the topic, the homework, the readings?
Well, don't hesitate to reach out to me. Let me go ahead and put my slide deck back up too, if you do have questions. And it was my pleasure to speak with you today, and I hope I hope you have enjoyed your trip through regulatory affairs so far, and I'm excited that there are so many of you graduating, because we need more regulatory affairs professionals in our region. So stay here and establish great careers for yourself, and let's build our ecosystem together.
Thank you very much.
Thank you
You
so
are
much.
more than welcome.
Thank you very much.
Thank you so much. And all of you students, enjoy the rest of your evening. Start the homework if you want, or relax for a little bit. But we'll see you next week. And Katrina, thank you so much. This is a tough topic to find the right individual to present it. I couldn't have asked for more than what you have done this evening. It was really great, and thank you for spending. And I'll be following up with you. So much appreciated.
Awesome.
Thank you.
It's been